Transport of substances through cell membranes is one of the mother concepts a biologist should know, because we know that what delineates cells is the plasma membrane. Plasma membrane, which we already discussed that it is a phospholipid bilayer. Now, let us consider this side extracellular fluid, this side intracellular fluid, meaning this one is outside the cell. In fact, this one you can call it tissue fluid. Tissue fluid, the immediate fluid outside the cell. Now this one here is the cytoplasm. So materials need to enter the cell or to leave the cell. So that is our discussion today. Now, before we get to know how materials enter or leave cells, we need to first of all know which materials are those ones that have to enter or leave cells. First of all, we can think of drugs. Drugs, if you have ever gotten a disease like malaria and you're given drugs like Coartem or whatever, these drugs have to enter cells. But also, metabolic wastes, they have to leave cells. We can cite examples: CO2, we can cite urea, we can cite uric acid, etc. These metabolic wastes, they are formed after biochemical processes occurring inside cells. For example, respiration, the ornithine cycle, and very many other processes that produce wastes. These wastes have to leave cells. Another example, we can give materials that can enter or leave cells. We can think of hormones. Hormones are produced by cells called glands, and they have to be excreted and released to go to target tissues. For example, cells of the pituitary gland, cells of the thyroid gland, they have to release hormones which will go and target faraway tissues at faraway places within the body. So these hormones have to leave cells, but also they have to enter cells where they're going to act to carry out action. Of course, some don't enter, but some do enter. We can think of proteins, both structural proteins. We can think of enzymes. Those ones are non-structural, but they are very important because they catalyze biochemical processes. Enzymes. We can think of antibodies, proteins of the complement system, etc. These ones are very important. However, the hormones we mentioned, some are proteins in nature, some are not proteins. Some hormones are steroids, some are proteins. Some are lipid-based, some are protein-based. For example, oestrogen and testosterone are lipid-based, are steroid hormones, but hormones like insulin and what have you, those are protein-based hormones. Steroids will enter easily. We shall get to know the reasons as to why. Then protein hormones may not manage to enter due to some factors we shall see. So these materials have to enter or leave cells. We can think of materials like oxygen. It's very important. Like water. These materials have to enter or leave cells. For example, during photosynthesis, oxygen is liberated during the light dependent stage of photosynthesis, but this oxygen will be released out of those cells, the pulsed mesophyll cells, where photosynthesis occurs. But also remember, metabolic water is made during aerobic respiration, which has to be released out of cells as a metabolic product. Therefore, those are some of the materials that we can cite. Very many materials have to enter or leave cells. So that is why, including drugs, drugs, for example, pharmaceuticals, pharmaceuticals, we can think of vaccines, etc. We can't mention and exhaust them, but very many materials have to enter olive cells. So today's discussion is on how these materials enter olive cells. Okay. Now, there are different methods by which materials enter olive cells via the plasma membrane. There are different methods. Now, these arrows you see, some are double way, some are one way. They represent particular processes by which materials enter olive cells. Now, A and B, that is the simple diffusion. A and B is a simple diffusion. Here materials move along their concentration gradient from where they are highly concentrated to where they are less concentrated. So both of them are simple diffusion. Category A, how do we tell simple diffusion being two-way sided? Two-way, that is simple diffusion. That is diffusion, two-way diffusion. But look, category A, it is through the phospholipid bilayer. These materials are passing through the phospholipid bilayer, category A. They pass through phospholipid bilayer. But category B, they are passing through a protein channel. This is a protein channel. This is a protein channel because it contains a channel, meaning this substance B, for it to cross via this membrane, it cannot pass through the phospholipid bilayer, but it has to pass through the protein channel because of some reasons we are yet to discuss. So A and B are simple diffusion. Okay. Now category C is facilitated diffusion. Category C is facilitated diffusion. Category C, unlike a B, where you see that there is a channel. In C, there is no channel. But a protein just changes its configuration. A transport protein just changes its configuration. Okay? And this is a transport protein here. This is a transport protein. It just changes its configuration and takes substances either outside or to the inside of the cell. So it is facilitated diffusion. Okay. Category D. Now, look, we have agreed that A, B, and C, all of them are diffusion. All of them belong to a category of transport called diffusion. Category D, that one is one way. Being one way, we are going to combine. Being one way, let's combine D and E. All those ones active transport. All those ones belong to what we call active transport. D and E is active transport. This type of transport, energy is expended. Energy is expended. In this category of transport, energy is utilized. So there is a consumption of energy in form of ATP for materials to be transported. I want you to note that it is one way. This protein here transports something from the extracellular fluid to the intracellular fluid. This one here from the intracellular fluid to the extracellular fluid. So such proteins, these two proteins here, these two proteins there We call them carrier proteins. Carrier proteins. But also, this protein here, which just changed, this protein here for facilitated diffusion is also carrier protein. Carrier protein. So how do they differ from these other proteins, like a protein for transport B, for simple diffusion category B? Is that for them they just change configuration. They have no channels. So let me hope you've understand that one very well. Now, we are going to have another video where we are going to break category per category. Thank you for watching.